In lecture 3, we are going to be looking at the immune system. We're going to look at the different types of defense we have in the body against pathogens. Some we are born with, some we have to develop throughout our lifetime. And we're going to look at things like vaccinations and some of the ethical concerns surrounding them as well. So immunity is basically your ability to fight off foreign pathogens. So if there's a bacteria that has entered your system, you have to have a way of dealing with it. So that's the job of your immune system. And there are two branches of immunity. There is what we call innate immunity. So innate immunity is something that you are born with. Okay, it's the inherited immunity that helps you defend the body against general infection. Okay, so this innate immunity is not designed to fight a specific pathogen. It's just kind of like a broad defense against a wide range of general infections. If you come down with something that's really specific, like chicken pox, for example, like the cold, then innate immunity is not very good at dealing with that. So that's when we're going to need something called the adaptive immunity. So adaptive immunity is something that is very specific, and you're not born with the ability to fight these specific pathogens. You have to encounter them first before you are able to create a tailored response to it. So adaptive immunity is also known as specific immunity. Specific immunity, or because you have to get it as opposed to be born with it, is sometimes called acquired immunity. Acquired immunity. So there are various mechanisms that are used by innate immunity to help us defend against a wide range of pathogens. They are listed here, and we're going to go into more details in the upcoming slides. And then in the adaptive immunity, the specific one, the big guns, so to speak, that involves producing specialized T cells and B cells, and you might remember learning about these in the previous lecture, as well as production of antibodies. So that would be the second part of the lecture. So let's start with innate immunity. The first line of defense against pathogen is actually the innate immunity, and one of the things that are involved in the innate immunity is the use of physical barriers. So the best defense is basically to keep all the harmful stuff outside of the body. Don't let them come in. And so we have built-in barriers, both physical barriers and chemical barriers within the body that help us prevent infection by microbes. So an example of a physical barrier would be intact skin. Your skin is actually very, very important. By having unbroken skin, it actually is quite an effective physical barrier that would keep things out of the body. Your skin actually has like a waxy coating to it. The keratin actually prevents things from penetrating through the skin. So, you know, if you have a cut, that's why you should like put a band-aid on or something. So you know the bacteria is not going to be able to enter your wound through the cut. Also, people who suffer from severe burns, they might have to be put in an isolation chamber where the environment is sterile, because you know if you have suffer from a burn that occurs on a large proportion of the body, then you're very vulnerable, you're very exposed to airborne pathogens. So that's why they need to be isolated. Where skin is not possible, the substitute for that is mucous membrane. This would be found, for example, on the inside of your mouth, found in the respiratory tract, digestive tract, reproductive tracts, as well as urinary tracts. So the mucous membrane serves the same purpose as the skin. It's just that it doesn't make sense to put skin in these areas. So you know the alternative to it is the mucous membrane. Finally, we have what's called ciliated cells, and in your trachea, in your upper respiratory tract, you would have these cells that have little hair on it. So these cilia will keep on sweeping up the mucus and sweep up any trapped particles up the throat, so you can kind of spit them out, right? So that way it prevents the colonization of harmful microbes. We also have something called normal flora, and normal flora are these beneficial microorganisms that lives on the surface of our body. So you will find these microorganisms on the skin, you will find them in the intestinal lining, and they actually have mutualistic relationship with us. We kind of give them a place to live, and in turns they offer some kind of protection for us. And the way they protect us is that they actually take up so much space in the body that it leaves no room for the harmful pathogens to attach to, and so they're not going to be able to gain access to our body. So having a good normal flora is a good thing. Now, if you take antibiotics, for example, and you read the the little pamphlet that comes with the drug, you will see that sometimes the side effects of antibiotics is diarrhea, and the reason for that is the antibiotics does not distinguish between good bacteria and bad bacteria. So when you take the antibiotics to treat whatever infection you have, it would also kill off some of your normal flora. So once the normal flora is depleted in the intestinal lining, now there are space that are being freed up, and these space can be occupied by harmful bacteria that could give you the diarrhea. But that's okay, because when you lose your normal flora, you can gain them back. People can eat probiotic yogurt, for example, right? There are these good bacteria in the yogurt that will help replenish the normal flora in your gut. Now, they are supposed to be beneficial for us, but sometimes the normal flora can be causing what we call opportunistic infection. So if there is a breach in the barrier, it's possible that these normal flora can go deeper into our body and cause some kind of infection. So here are some examples of normal floras. Here we have Staphylococcus epidermidis. So the bacteria name usually comes with two parts, just like we have first name and last name, and the names kind of tells you what kind of problems they might cause. So here we have another one, right, that causes acne, and we have Staphylococcus aureus. So we don't usually say the full name. You probably have heard of E. coli, right? So E. coli. So E actually stands for like a long name. So here Staphylococcus aureus, people usually just say S aureus. So S. aureus is particularly concerning for healthcare worker because there is something called MRSA, right? MRSA. And if you do a quick Google search, there are tons of news about MRSA, on a monthly basis, I would say. So what is MRSA? MRSA stands for methicillin resistant S. aureus. Okay, so methicillin is a very strong antibiotics. It's one of the strongest one that we have, and because of antibiotic misuse, bacteria have developed resistance to these things. So what happens is, if I have a bacteria here, a regular S. aureus, and then I have another one that is going to be resistant to the methicillin. I'm just going to put some hairs on it to show that it's resistant. What happens is, if these two come together, then it could pass the resistance onto the non-resistant one and convert it into resistant. So what that means is, if you you have S. aureus on your body, and if you go get contracted with the MRSA, then what happens is it will convert your good ones into resistant one, and that's particularly concerning in healthcare setting, because if you come down with this bug, the MRSA, then even the strongest antibiotics is not going to help you. And if you, on top of that, if you're fighting another illnesses, then your chance of recovery is going to be affected. So normal flora typically good for us, protects us, take up all the space so the bad guys can attach. But in some cases, they can cause opportunistic infection. So another type of barrier is chemical barriers. Chemical barriers include what we call a lysozyme. So lysozyme is an antibacterial enzyme that are found in sweat, saliva, and tears. So every time you blink your eyes, for example, the tears will be replenished on the surface of the eye, and that basically kills off any airborne bacteria that landed on your eyes. If you rub your eyes a lot, for example, right, then this kind of mechanism would help you keep the surface of the eye clean. Another example of chemical barrier is the presence of acid in the stomach. So your stomach has hydrochloric acid in it, and the pH is very low. It's about 1.5 on a 14-point scale. So the lower the pH, the more acidic it is. And that's a good place to have acid because the stuff we ingest are probably teeming with microorganisms from the air. So by having the acid there, it will kill off as much as we can. The vagina is also a little bit acidic, and that's important for inhibiting bacterial growth in that area. Now, even the best physical barrier is not going to be able to keep things out all the time, right? We are going to have cuts, for example, then there is going to be a breach in the skin, and the pathogens will now be able to enter the body. So if it enters the body, one of the first local response to tissue damage or infection is inflammatory response. And this is initiated by the release of histamine from what we call a mast cell. So you might remember basophil, right? Basophil, we learned about basophil in the previous lecture. Basophil also is responsible for the inflammatory response. So the difference between the mast cell and the basophil is that the basophil tends to be circulating throughout the body, while the mast cells, they are anchored to tissue. Okay, so mast cells are responsible for more like a localized type of inflammatory response, whereas basophil is going to be more of a systemic type of inflammatory response. So let's take a look at this in a note. When you have a cut, or if you have an infection, then the mast cell is going to release histamine. Now, histamine is the key chemical that causes the inflammatory response, and it's going to have two effects on the blood vessels. Number one, histamine is going to cause vasodilation. Vasodilation. So vasodilation is basically means that the blood vessel is going to become bigger. It's going to increase in diameter, and this is going to allow for an increase in blood flow. By having an increase in blood flow, what you're going to be able to do is you're going to be able to recruit more WBC, white blood cells, right? Because you're having a cut, you're having an infection, it makes sense to recruit more WBC to the site of infection so you can deal with it. And you can also recruit more more plasma protein. So you might need to have some. You might need to have some clotting factors, right? Since you have a cut, so maybe some prothrombin, maybe some fibrinogen. You are gonna maybe bring some generic antibodies. And there are a few other things that we're going to learn later that is important for the defense. But the key idea here is the histamine will cause vasodilation, make the blood vessels bigger, like I said, and that will increase blood flow. And the purpose of that is to bring more white cells and plasma protein to the site of injury. Now, the side effects of this, the side effects of vasodilation is because you're bringing more blood to the area, so you are going to have two side effects from that. Number one, it's going to be red. So there's redness, and is going to feel warm. Okay, warmth. Okay, so these are two of the characteristics of inflammation. Now, after recruiting the white blood cells and the plasma protein to the site of injury, we want them to be able to exit the capillaries. So typically capillaries, as you know, are already quite leaky, but because we have an increase in these things, they might not be able to get out fast enough. So the second effect of histamine on your blood vessel is it will increase permeability. Okay, so not only are their vessels bigger, but now they are like extra leaky. Okay, so by having extra leaky walls, the white cells can now exit. Okay, and then if you have proteins like antibodies and clotting factors, they could exit into the tissue as well. So that would allow WBC and plasma protein PP to exit blood vessel into the affected tissue. Okay, so what's the side effect? What's the side effect for this? So the side effects, because it's extra leaky now. Not only are the white cells and the plasma protein leaking, but fluids will be leaking as well. So there is going to be swelling. Okay, and all that extra fluid that are oozing out is going to be pressing on your nerve ending, and it's going to give you that distinct throbbing pain associated with inflammation. Okay, so once again, the purpose of inflammation is to recruit more white cells and plasma protein to the site of injury, so you can clear the infection, you can start wound healing, and the characteristics of inflammation are listed here: redness, warmth, swelling, pain, and they're actually side effects of what histamine is trying to do to your blood vessel. So that is the inflammatory response. Now back to the PowerPoint slides. Here is a blood vessel, right here. There is a blood vessel, and over here, you kind of just have to trust me that this is a mast cell. Okay, mast cell, right? So if you have a injury, some kind of thumbtack, then what happens is the mast cell will now release histamine, and the histamine will act on the blood vessel, right, causing it to become bigger, right, vasodilation, and it will make it more leaky. So then when the WBC and the plasma protein comes, they will be able to leak out and deal with the infection in the tissue. Here is a good summary of what's happening. So I encourage you to read that on your own and see the whole process step by step. I mean, there are other stuff that we haven't talked about yet. We will in the upcoming slides. So maybe come back to this slide afterwards and just use it as a study tool. Now, in case you forgot, we are basically talking about the different mechanisms for innate immunity, right? These are immunity that you're born with, and is used against a wide range of pathogens. They are not tailored. Okay, so we talk about barriers, keeping things out, and if the barriers fails, the pathogens, the bacteria came in, then we have an inflammatory response. And now another mechanism we have is phagocytosis. You probably remember that, if you have a phagocyte here, and there's a bacteria, then the phagocyte is basically going to eat up the bacteria, kind of like Pac-Man, and then you're gonna do phagocytosis, right? And that's how you get rid of the bacteria. In lecture 2, we learned about 2 examples of phagocytes, right? We have the neutrophils, right? The most abundant white cells you have, and we have the macrophage, which are came from monocytes. So these are both phagocytes. Then they could go around and eat stuff. Now, how do they know what to eat, right? Well, the short answer to that is they would eat up anything with what's called PAMs on it. Okay, so PAMs stands for pathogen associated molecular patterns. Okay, so these are a group of antigens, kind of like barcodes that are on the surface of pathogens, and if the phagocyte sees the PAMs on a target, then they're just going to go ahead and eat them. So here we have the blue is a phagocyte, and the yellow is something with PAMs on it, and so you can see that it's basically eating it up. In fact, you can tell it already ate one of those things. So typically the phagocytes they have quite a big of an appetite. They could eat up to hundreds of bacteria before dying. So sometimes the macrophage come, and then they realize there are too many bacteria and they cannot deal with it themselves, so they would call for reinforcement. And the way to do that is to release a chemical signal called the colony stimulating factor. Okay, so you might remember from last lecture that in the bone marrow there are these stem cells, and the stem cells could become red blood cells. They could become platelets. And they could become the different kinds of white cells, right? And all of these lineage would be triggered by the presence of certain types of chemical signals. So when macrophages release colony stimulating factors, then these factors is going to tell the stem cells in the bone marrow to make more white cells. Okay, so particularly they're going to make more neutrophils, right? More monocytes, so that they could be recruited to the site of infection to help clear the infection. As the infection is being overcome, some of the phagocytes will die. In fact, the dead phagocytes, along with the dead tissue cells and dead bacteria, even living white cells and possibly living bacteria, they sometimes form these pus. Right? Pus are the clear yellowish, sometimes fluid that oozes out from your wound. A little bit of pus is okay. It means that your wound is healing. But if the pus is not being drained and it's kind of accumulating in the body, then that could cause trouble, and you might have to drain it manually, like at the hospital, for example. So the presence of pus indicates that the body is destroying dead tissue cells, and because it potentially contains live bacteria, pus is considered as an infectious agent. Okay, so make sure you don't touch people's pus without proper protective gear. Now, related to phagocytes are these things called natural killer cells, okay, NK cells. Natural killer cells, they will kill virus-infected cells, and sometimes, with limited capability, kill tumor cells. So just a little bit of virus biology to help you review. So here we have a virus. In order for the virus to replicate, what they must do is they would inject the DNA into a host. So in this case, the host would be like your cells. And then the infected hosts will now produce virus parts, and the virus parts will assemble, and then the virus will burst out from the cell, and then they will go infect other cells, right? So what happens is when your cells are infected with the virus, it actually will put some chemical signal on the surface. Okay, it will put some chemical signals on the surface that tells other cells that I'm infected. Okay, and so please destroy me before I turn into a virus factory. It's kind of like a zombie movie, right? Like someone gets bitten, they're like, please put me out of my misery before I turn into zombie. So that's what the NK cell is for. The NK cell will come along and see that this cell is being infected, and they would destroy it. So NK cells is a little bit different than phagocytes in that they don't kill the target by engulfing them. They don't eat them up. They have a way to just touch the target, and then the target will die. We're not going to go into too much detail for it, but suffice to say, they use cell-cell contact to destroy the target. So similar things can happen to tumor cells. But is one of those things that when it's working, we're not aware of it, because then you wouldn't get a tumor. But once this fails, then cancer is possible. So that's the third type of defense, with the phagocytes and the natural killer cell. Next, we have something called complement proteins. Complement proteins are basically plasma proteins. There are many different types of complement proteins, and their names are kind of boring, right? C1q, C1r, so on and so forth, and they are mostly made in the liver, and when the body is not fighting the pathogen, most of these proteins are going to remain inactive. Okay, so it's very similar to the concept of clotting factors, right? Clotting proteins, right? Like the prothrombin and the fibrinogen, they are not active until you need them, right? So in the same sense here, if you don't have an infection, there's no point turning on all these complement proteins. So once you have an infection, though, you will want to turn on these complement proteins, and to do that, it's basically a cascade of chemical reactions, right? So you would activate, for example, C1QRS would activate C4, and C4 would in turn activate C2, and so on and so forth. So it's like a domino effect, right? The activation of one complement proteins will result in the activation of the next. So these complement proteins, their main job is to synergize or to enhance other non-specific defense. So it's going to help with inflammation, for example. It's going to help with phagocytosis, right? So there are many different functions for the complement proteins. We are just going to look at three main ones. So number one, complement proteins will help recruit more WBC. As you might remember, the purpose of inflammation is to recruit more WBC and to recruit more plasma protein, right? So complement protein is going to help synergize that, is going to help enhance that that function. So if you think about a new WBC, new white blood cells coming out from the bone marrow, how am I supposed to know where to go? How am I supposed to know where the site of infection is, right? It would take a long time to just randomly circulate through the body and, eventually getting to the infected site. So complement kind of solves that problem. Complement proteins will leave behind chemical trails for the WBC to follow, kind of like breadcrumbs, right? For the white cells. So the white cells will just continue to follow that chemical trail. Eventually, it will lead to the site of infection. So here, we have the splinter here, and all these are complement signal, and all the phagocytes are basically attracted to these chemical signal so that they know which way to go. Okay, so that's the first function, recruiting more WBC. Second function is that complement protein can coat the targets. Okay, by coating the target cell with complement protein, you will be the phagocytes will be able to identify them easier. Okay, so even if the let's say this is the tissue, right? And there are a lot of cells here, and then there might be some bacteria. How does the phagocyte know which one to engulf? sure, there is the PAM, but they still have to check the cells, right, to see which one has the PAMs or not. By coding them, it actually highlights them and makes it much easier for the phagocytes to identify them. So it's kind of like, you know, finding Waldo, right? I can ask you to look for Waldo. Okay, you would be like the phagocytes, and you know, Waldo in this case would be like the bacteria or something. So you're looking all over the place, and you know, eventually you're gonna find them. But you know, it's gonna take you some time. But with the help of complement, right? Complement proteins will put a big halo around the target. Okay, so when you look at the picture, immediately you'll be like, oh, there's Waldo, right? So in the same sense, right? If you highlight the bacteria, then the phagocytes will be able to identify the targets much quicker, and so you can make a beeline to the target, eat it, and hopefully that will get rid of the infection much quicker. And the third thing that complement proteins can do is that they can form these things called membrane attack complexes, or MAC. So these MAC will insert holes in the bacteria and cause them to leak all over the place and die. So the other two mechanisms that we talk about, the chemical trails and the tagging the targets, those are kind of strategies to enhance other immune defenses. But here, this ability to form the MAC, it's kind of on the offense, right? It punches holes in the bacteria and causes them to die. Sometimes, on top of everything else that we've talked about, the inflammation, the complement proteins, the phagocytosis, the natural killer cells, that might not be enough. So we might have a fever in response to the infection. So fever is a temporary increase in body temperature. Normal body temperature is 37 degrees Celsius or 98.6 Fahrenheit. And when you have an infection, there is a group of chemicals called prostaglandin. Okay, and the prostaglandin will act on the temperature controlling region in the brain called the hypothalamus, and that in turn will increase the body temperature. So here is the hypothalamus. We're going to learn that in the nervous system unit. And by having a fever, it helps with the immune defense by increasing enzymatic activity. And enzymatic activities are basically chemical reactions, right? And they tend to occur a little bit faster when the temperature is higher. So by doing that, you can increase the rate of complement activation, for example, and you can speed up metabolism, right? So that helps with the wound healing process, increase the rate of tissue repairing, and phagocytic activity. So having a fever, if it's not super high, right, having a low-grade fever is like pressing the fast-forward button when you watch a movie, right? So everything just goes a little bit faster. And typically, if you just have a low-grade fever, you shouldn't take any medication. You should just let it run its course, it's supposed to be a defense mechanism that helps you get better quicker. And that's it for innate immunity for now.